So Joe Hacia is going to talk about peroxisomal mouse models, and he'll give us an overview and describe the relation to ALD at large. There we go.
Thanks. Visually challenged over here.
No problem.
All right, thank you very much for the opportunity to speak here today. My name is Joe Hacia. I'm on the faculty. I'm a geneticist at the University of Southern California, and this is my first time to ALD Connect. And thank you, Florian. All right, I got my stature. It's good. Now, I also have to remember that Florian, when I gave a talk at the GLIA conference, encouraged me to have some joke slides. So I think he's going to open up a Pandora's box. So just remember that. Again, this is my first time to ALD Connect, and it really is a privilege and honor to be here. And hopefully this talk is appropriate because it is really about human connections and research connections. And the objectives is going to be, I'm going to be mercifully brief in the first one of describing the fundamental roles of peroxisomes in human health and in rare and common diseases as a way of getting to the heart of the talk, which is explaining the aims of a recently funded NIH R24 mouse models for peroxisome research grant with the Jackson Laboratory. And then we'll have discussions about opportunities for the ALD scientific community to engage in peroxisome-related research and make use of these newly developed mouse and monoclonal antibody resources. And like most conferences, a lot of the work gets done over drinks or over dinner afterwards. So I'm looking forward to many discussions. So just as a brief overview about the basics of peroxisomes, we think of them as being metabolic organelles. They're present in almost all eukaryotic cells. And most of the work or the summary points that I've put on the slide is actually due to the work of many of the investigators here in this room today. We've already focused on their role in the metabolism, potentially toxic fats in the brain, such as very long chain fatty acids. Not well as discussed, but often comes up in meetings of neurology of peroxisomal disorders, is their role in generating plasmalogens, which are ether phospholipids that are very abundant in the myelin sheath and important for structure and function. But in addition, they're involved in metabolism of many other biomedically or metabolically active molecules that are involved in human health. And these include bile acids and lung surfactant, and also in the catabolism of amino acids, glyoxylate, and polyamines. So these are probably words that for the medical students or people who have gone through medical school in year one have heard and very rapidly forgot because we don't really hear about them too much in the medical community, but they really are important for the structure and functions of many of our cells and tissues in our body. And they are a major hub of cell signaling pathways involving reactive oxygen species. So all told, we know that there's many lines of evidence to say peroxisomes are involved in the functions of most major organ systems, even though we're talking about the nervous system here today, and we also talk about indirectly the immune system, but they're also involved in the structure and function of the reproductive system, of the cardiovascular system, and so on. So I'm giving this as an introduction because when we start talking about some of our mouse model resources, I want you to let you know that we're not limited to making models of CNS disorders, but any disorder or any biological process that the peroxisome is involved in the human body. So just to give you a little brief of an overview of the players in peroxisome structure and function, we think about peroxisome biogenesis, or how they're made, as being in two parts. There's a de novo formation of them from the actual budding off of vesicles from the endoplasmic reticulum to form pre-peroxisomes with some vesicles that are budded off from the mitochondria. traditionally we think of it primarily as obtaining membrane from the endoplasmic reticulum. And then there's a process by which these peroxisomes mature, where they acquire peroxisome membrane proteins. And there's a number of them we'll be talking about in a moment. PEX proteins or peroxins that are involved in the assembly of peroxisomes, as well as transporters like ABCD1. And then the next step is to get the enzymes or the workers that actually have to be, that are produced in the cytoplasm of the cell into the peroxisome matrix where they're actually going to be doing all the work. So this is the, an overview of how the peroxisomes are generated. And let's just go into slightly more detail so we could dive a little bit more into the cast of characters. I mentioned that there's a number of proteins or enzymes that are required for peroxisome functions. They're metabolic machines, and they require enzymes for their structure and function. So these enzymes are actually made in the cytoplasm, and they have appended to them, either at their N or C terminus, peroxisome targeting signals, short amino acid stretches that basically convey the information to the cell that that this particular enzyme or protein has to go into the peroxisome itself. So there's a whole series of proteins that are involved in getting those enzymes into the peroxisome matrix. Now, the nice part about them, they're all encoded by PEX genes. So anytime you hear the word PEX gene, you know that's going to be involved in peroxisome assembly. And they're also encoding peroxin proteins. Now, the bad news is that they were named based upon the order in which they were discovered. So it's really a random collection of numbers, right? So you'll see PEX peroxin 5, 7, 2, 10, and 12. And my good friend Paul Watkins will call it pexology. And for those who have used to look at the science fiction series Lost, from the many years ago, there was a character, Curly, who had many numbers that were going through his mind. That's basically the world of peroxisome biology. But the bottom line is all these proteins, which are involved in dropping off these passengers, these enzymes, and deposit them into the peroxisome matrix, all these proteins are important for the assembly of peroxisomes, and having defects in any one of them, or defects in both copies of the genes, one from your biological mom, one from your biological dad, can wreak havoc on peroxisome biogenesis or assembly. So, the theme for this particular meeting, or one of the themes outside of family and community, is these rare peroxisomal disorders. And they could be divided into two different categories. The peroxisome biogenesis disorders, as I referred to, are diseases where there's rare diseases where there's defects in these PEX genes. And as a result of this, the peroxisomes are just not assembling properly. The enzymes are not getting into the peroxisome itself. Sort of like the equivalent of, in this room, these doors were not working and there'd be nobody entering the meeting hall. All right. So they run into two different categories, which I won't dive into too much depth, but one is called Zellweger spectrum disorder. Another one is rhizomelic chondrodysplasia punctata. Then there's the single transporter and enzyme deficiencies. And this is really the theme of this meeting, of adrenoleukodystrophy as being a single transporter defect. Just one particular peroxisome membrane protein is defective. And as a result of this, as was described by Florian earlier on, that the peroxisomes are actually not importing the very long chain acids into the matrix where they are actually catabolized or broken down. But there's a whole series of other single enzyme deficiencies. There's about 12 of them, where it's essentially just one protein that's involved in an enzymatic pathway or a metabolic pathway is defective in the peroxisome. The peroxisome is assembled, but there's only one defect. And I've shown pictures of two of the leaders of a group called Global Dare, and these individuals have a disease called adult Refsum disease, and they just have the inability to break down one type of toxic fatty acid called phytanic acid, and as a result, they gradually lose vision and they have a peripheral neuropathy. And I'm involved in a number of projects investigating that with taking the lead, the investigators at the Kennedy Krieger Institute, and also with Florian. So the other diseases that I will just briefly introduce for the just to give you an idea of where the mouse models have become of great value to our community, is Zellweger spectrum disorder. This is one of the subtypes of a peroxisome biogenesis disorder. And these are autosomal recessive, and it's ultra rare, about 1 in 50,000 births in the United States. And there's around 200 registered patients in the United States. And it's a spectrum disorder that can have a variety of phenotypes or manifestations of disease in the clinic. And this is really driven primarily by the severity of the mutation in the patient, whether or not both copies of the gene of interest have any residual activity. So if there's some residual activity in one of these PEX genes that in the mutated state, then the peroxisomes can form a little bit more efficiently, and then they kind of have some function. And as a result of that, the patients can live longer. So when I see some of my favorite people on the slide, this is Nancy Braverman, who's one of my partners in crime and research. She's over at the McGill University, a physician scientist with a family whose daughter has a more mild form of peroxisome biogenesis disorder. And we know that when we say the word mild, it's always, it's all relative. And so this little girl here is able to, to speak and communicate with us. And in fact, she can actually translate for her parents from Spanish. So she's far more adroit at multiple languages than I am. Then you could see on the other end of the spectrum, you could have children who live to only 1 to 2 years of age if they have the most severe forms of the disease. So there's a lot of different genes that are involved. There's what we call locus heterogeneity, where you can have mutations in both copies of the PEX1 gene, 6, 10, 12, and so on, and they can give rise to very similar medical conditions or medical phenotypes. And that's really driven by the amount of residual activity in the gene itself, about how severe it is. And so if you look at this PEX1 G843D allele, this is something that Troy Lund also studies as well as his lab. This is a founder allele, meaning about it's a higher prevalence in the patient population. And it's conducive with a longer lifespan because it actually has some partial activity. And as a result of this, about 30% of patients have this particular allele, and it's conducive with longer-term survival, where if you have 2 of these alleles, you can grow to adulthood, and then there's a gradual loss of vision and hearing, but they can be cognitively intact, many of these individuals. And so this is more of a degenerative type of disorder. So several years ago now, it's actually going back to 2014, if my eyesight can read the slide, we published a paper as part of a group on making a mouse model, which was really funded through a parent organization, and it was one of the pictures of the children I showed in the prior slide. It's very near and dear to my heart. The Ann Park and Matt Hopkins are a family who lost their son Peter to a peroxisome biogenesis disorder, and they did some fundraising to actually pay for this mouse model. And so they're near and dear to my heart, and this is something that has motivated me to work in the mouse world. And so at the time, as I was joining the team, not to brag, but my mouse acumen was really being an annual pass holder at Disneyland. But nevertheless, I took my nerdy talents of bioinformatics to try to help along with the project. And those particular mice, you could see here one of the healthy litter mates versus the mouse model itself that has 2 copies of this common variant in the population. You can see that they're smaller in size. They've been well characterized to show retinal degeneration and cholestatic or liver disease. They actually have a bile acid deficiency, so it's a digestive problem. And those mice are very good for studying those biological manifestations of the disease. But it's always a coin toss or a throw of the dice whether or not your mouse model will recapitulate all aspects of the disease. So very similar to the problems with the ALD mouse models, our mouse model does not show any CNS phenotypes. So that makes it very difficult for us to study any pathology or treatments. So that really just shows us, the power as well as the drawbacks of some of these animal models. So as part of my part of the team, if there's one thing I've been able to do is help put together some teams. And we started working with a group over at the Jackson Laboratory. And so the way I think of it, if you hear the word Starbucks, you think coffee. If you think about Jackson Laboratory in Bar Harbor, Maine, you think mice. They're one of the premier centers for making genetically engineered mice. And so they started working on this model to try to optimize it. And there's all these issues of genetic backgrounds. We talk about strains of mice. I think about them as breeds of dogs almost. I'm showing my New Jersey accent over there. And depending upon the breed or the strain of the animal, you could have differences also in the phenotype in terms of its survival. So they've done a lot of work optimizing the system for us. And what the two points that I've learned here is the importance of humility about working within teams and appreciating that there's experts out there who can solve problems that would be far beyond the scope of what I can be able to do. And also the power of what these families have done in our community. And I go to a lot of the fundraisers, and it really struck me that with all these different genes and all these different alleles that could give rise to these peroxisomal disorders, it feels really unfair that the burden is on families to do all this fundraising. And not everybody has the wherewithal. And we just heard about some social justice issues. We can't expect everybody to be able to drop everything, put on these dinner gala events and fun runs and raise $50,000 to $100,000 and pay for a mouse model to be made. So I wanted to see if there was any way to address that. And that really brings a lot of meaning to my life. So I became aware of an NIH granting program. It's for a program for making research resources for the development of animal models and related materials. So I've been on NIH study section panels like this, and they tend to fund, big picture things like we're going to make general fly models for any disease you can imagine, or worm or zebrafish. And those are the critters or the organisms that are very popular in the biomedical research community. But occasionally they have quirky types of projects that get funded as well. And there's nothing more quirky than peroxisomes or myself. So as a result of this, we looked at this program, and I said, well, we can be able to try to make peroxisomal animal models or mouse models of disease, and we can try to be able to share this with the biomedical research community to try to lower the bar for people to come into the field. Sadly, it's a very poorly funded field in and of itself, the mouse model world for peroxisome biology. So we can be able to get you potentially animals, but we can't actually pay for the experiments with them. But nevertheless, we open the door. So by working with the group at the Jackson Labs, they did an extraordinarily act of kindness for us. They made at least over 20 different animal models for free for us as preliminary data. So that is something that, is probably understated, but behind the scenes, we really have some really genuinely saintly individuals who are working with us. And one of them is a person by the name of Kat Lutz, who's one of the directors of their rare disease program. And many of you may have heard her because she's worked with many rare disease communities. And for example, she made a mouse model for ALD where she placed the ABCD1 mutation to have a null allele with no activity on a background that is conducive for hematopoietic stem cell transplant. So you could be able to transplant human cells into this particular animal, and they will engraft. And this is a non-skid animal, and it's actually commercially available now. And she did that for free for us. And I know a number of investigators in this room have started to explore this. So that was part of the preliminary data. And my good friend here, Paul Watkins, also showed the spirit of sharing in that he deposited some of his animal models for ALD, the bubblegum mutant, into the JAX repository, which is important to deposit these animal models once they're made in a person's lab, so they just don't get lost when a person may retire or they may have some problem with their colony. That way it's kept in perpetuity. So just to get to the heart of the matter here with our grant, it was almost a sign of the apocalypse to me. But after 2 years, I actually got an NIH grant, so I was very, very happy about this. And I was more happy that for the families, because to me it was a sign of hope that maybe I can get back to the community a little bit and have families do fundraisers so they can be able to test potential therapies in these animals instead of having to generate them themselves from scratch. So we have 3 specific aims in this particular grant, and one is to consolidate, phenotype, distribute, and cryopreserve existing animals, and mouse models. Just as Paul deposited his mouse strains, we can now fund that to happen at JAX. And then 2, which is even more exciting, is to develop, phenotype, distribute, and cryopreserve new mouse models for peroxisomal disorders. And then finally, we're going to try to develop a monoclonal antibody resource to maximize the impact of these particular mouse models. So we use these monoclonal antibodies to basically track where different proteins are being made in the bodies of these animals, in tissue sections and in cells. So it's a 3-headed beast over here. I'm the lead PI on it, but there's 2 other PIs. Amir Zuberi is a genomic modeler and mouse modeler supreme over at the Jackson Laboratory. I mentioned my dear friend Nancy Braverman over at McGill. I should also give a shout out to the Kennedy Krieger Institute, which is going to be, and especially Ann Mosier, one of my favorite people on the planet, sitting there in the back, working on biochemical profiling of these animals. So the idea here in this, our goal here, if I could see here, is if you celebrate the season, Merry Christmas, because we could be able to give you some animal models for free. That's actually, it's pretty cool, isn't it? So the idea here in AIM1 is to, it's a 4-year grant of about $480,000 direct costs every year. So that's actually pretty good, and we can have the ability to renew it once. So if we show to the NIH that we actually had people buying into the resource, they may give us an extra 4 years of funding. So we're about 6 months in. So this is just the numbers of strains that we hope to import and a number of new strains that we hope to generate, as well as the antibody resources that we can be able to make and distribute to the community. So for the mouse model plans over here, we're trying to generate resources to generate global maps of peroxisome abundance and distribution. And actually, I just learned, even just like last month, that Troy was working on his mouse models where you can mark peroxisomes with a green fluorescent protein. So we're making similar models now that we're accepting nominations from the community. Instead of green, we could use the whole Crayola box of colors which will enable technologies that will enable us to address a lot of therapeutically relevant hypotheses. In addition, we're interested in resources to define therapeutic windows for degenerative disorders, meaning that we can have a transgene of interest. Let's just say we wanted to express ABCD1 and just turn it on in a shot in the cells, in the mouse, instead of having to design a virus to be able to integrate it, to be able to deliver it. We could engineer the mouse so that we can turn that gene on just by giving it a common drug called tamoxifen. And this is actually pretty useful. And we can either knock out a gene of interest or be able to overexpress a gene of interest on demand. This is common technologies that are now being applied for the peroxisome world. And then we are also interested in targeted therapy. So we want to work with patient-specific alleles. And again, that gets back to the idea of taking the burden off of families to have to do all the fundraising, that we can accept nominations from the field. If we could have researchers willing to use the mice, we can be able to work with them. The monoclonal antibodies, which has always been a bane of the existence of many researchers, developing these resources. ABCD2 is a big one that we've heard from the community, and we're going to try to attempt to make monoclonal antibodies against that particular protein. We recognize that we're going to allow people to try it and fail, so that doesn't mean we're necessarily going to succeed, but at least we're going to make an effort. And there's a couple other proteins, PEX proteins, that have been nominated by other investigators in the field. We're going to try to make them specific for the human, mouse, or cross-reactive forms of the proteins. We're also focusing in on specific applications. This is more technical for the biomedical researchers in the room. We're promising that our main attempt is to have them for Western blots. There are other applications called immunohistochemistry and immunoprecipitation that we would love for them to work, but we can't promise because sometimes the best the best intentions don't always come to pass, but this is what we're going to try to focus in on the Western blots. And we are soliciting new requests for upcoming years. So in the last, just to finish up, in the last two meetings, Florian has with erudite intentions, has quoted ancient philosophers to me. And one of them, you might remember, is this is going to be Virgil. So I don't know if Florian remembers a quote from Virgil. It had to do with the Trojan War. Well, I'm going to, let's see, I can educate you over here. It's called Beware of Geeks Bearing Gifts. Well, actually, it's Greeks, but in this case, me as a geek over here, over in Disneyland here during the holiday season. And so we're not giving out Trojan mice. They actually have their normal blood and guts inside of them. But the whole key here is it's all altruistic from all the investigators involved. We just want you to cite this resource in your papers and to do great science. We're not looking to get additional authors on papers or promote our own careers because ultimately this is the best things are done in teams. And with the idea of actually promoting science in this group, it would be near and dear to my heart, and especially if it can be able to help some of the families in this room. So the other Western philosopher that I will end up on is a quote from the wonderful Walt Disney that I only hope that we never lose sight of one thing, that it was all started by a mouse. These are my fabulous collaborators. I love the team here at the Kennedy Krieger Institute, and thank you for taking time to listen.